Hello, welcome to Area of Study 2 in Unit 4. So what we're going to do is we're going to talk about genetic engineering, we're going to talk about how we do it, we're going to talk about the enzymes used. But to begin with, I thought I'd just give you a little bit of an introduction into what is this thing called GE or GM or genetic engineering. So why would we want to change the DNA of an organism? So it's a question that you can ask yourself. What benefit is there? So thinking back, what does DNA do? Why is DNA important? DNA codes for proteins. Yes, so genes code for proteins. So if you change the DNA, you can change the proteins that are made, and that can therefore change a whole bunch of different factors in your body. So remembering back to what proteins do everything. Yeah, they form structural proteins like this. We have a lot of enzymes in our body that we use to break things down. We need proteins for antibodies, so our immune system. We need proteins for our hormones. So you can see that just by changing the DNA, you can change all of these functions within an organism. But what does it mean? What is DNA manipulation? So when we go into your notes, we should be on page 2, right here of the booklet called DNA Manipulation. And we're going through at the moment just a little bit of background, and then we'll launch into the study design dot points on enzymes and recombinant plasmids as vectors. So DNA manipulation is the same thing as genetic engineering. It's just essentially humans modifying DNA in order to make it do something different. That's it. So deliberate modification or changing of an organism's genes. You can also use GE for genetic engineering, and then GM means genetically modified. So this is an organism or something that has been genetically altered by the process of genetic engineering. So genetic engineering is the process, genetic modification is the product. So here are some of the uses of genetically modified organisms. One of the main ones that we're going to talk about is the manufacture of insulin. So insulin is very important. We've talked about this so much. So insulin is very important, particularly for diabetics that can't produce their own insulin. So we actually manufacture it and have since approximately the 1980s using genetic engineering. We also can use it to find out where genes are being expressed in the body. So by changing in this mouse in the middle here, by changing a gene to a fluorescent protein, they can actually then grow a mouse after they've changed the embryo and figure out where that gene is expressed in the human body by shining a UV light over the top. So pretty cool to make glow-in-the-dark mice. We can also really helpful for agriculture. We can manufacture crops that are resistant to disease. We can manufacture crops that have a higher yield. Yes, and this bit here, these are specific examples that you need to know for this study design. And we will come back to them in a little bit. So another thing that we're going to talk about here is just genetic engineering involves removing genes from one organism and inserting them into another organism. Now that can be either the same species or a different species, and we will come back into the nuance of what we call that later on. Now any organism that has had its DNA altered, we call that a genetically modified organism. So there are a couple of videos here. I'm going to link to those in playlist, and they're optional for you to watch, or we might watch them during class. So I've just got a little bit of a quiz for you coming up as to whether or not you think something has been genetically engineered or not. So there's lots of cool pictures that I'm about to show you. So here we have genetic engineering, perfect day for a banana fish. So I want you to pause after each one, have a think, and go, ooh, is that real or is that fake? So this guy over here, this grasshopper lizard, do you reckon he's real or he's fake? In addition, we also have this Oh, poor chimp dog. Think about it. Nah, they're both wrong. Yeah. So both of these organisms don't exist. They're just really cool examples of Photoshop. So let's do it again. We've got a blue rose and a purple tulip, or another kind of flower there that I'm looking at. So what do you think about these guys? All right, the one on the side there has just been coloured, either artificially using Photoshop, or you can do things like putting flowers in different coloured water and it will make them grow. Then in addition to that, the one on the left though was genetically engineered. So this blue rose was made by Suntory Flowers, which are the same people that produce alcohol like Midori. They're a Japanese company. And the black tulip is actually a dark purple colour, and they say that that one has actually been around since before the advent of genetic engineering. So have a look here. We've got some purple tomatoes and some very cool purple and orange carrots. So the purple and orange carrots are actually naturally occurring. So they've been around for a very long time. Prior to the advent of genetic engineering, but the purple tomatoes, or blackish purplish tomatoes, are a genetic modification. They were done to increase the levels of antioxidants. How cool is that? So they genetically modify them to have more antioxidants, which are better for our health, mean that they can get rid of free radicals and weird particles in our body. So now we've got two. We've got a mouse with an ear growing off the back and a glow-in-the-dark mouse. First one is literally just a makeup artist having fun with a mouse and shoving a ear onto the back and getting it to grow. Sorry, it's not a makeup artist. They did create the ear, but then they just transplanted it on the mouse so that it would have a blood supply. So it's not genetic engineering, it's just more of a lab process. Whereas a glow-in-the-dark mouse has been genetically engineered. They took a gene out of a jellyfish that makes it glow and then put it into a mouse and made it glow in the dark. Pretty cool. So here we've got some glow-in-the-dark zebrafish and some glow-in-the-dark jellyfish. Hopefully from the slide before, you had to think about that one. So the glow-in-the-dark zebrafish have actually taken the gene from the jellyfish, and been inserted into their own genome. Whereas the jellyfish, they just do this naturally. How cool is that? Some jellyfish can fluoresce. Okay, so we've got more glowing things. We've got a glowing tobacco plant, and then very cute little glowing snouts and, what are these called? Hooves of these pigs here. Now, both of these guys are genetically engineered. It's that same jellyfish gene. You'll hear a bit about jellyfish genes. They're very easy to manipulate, very easy to distill. So we take them out and insert them into things. And the fluorescent protein is really helpful for us to see where the protein is being expressed. So we often use a jellyfish glow-in-the-dark gene and insert it into things to see where things will end up in the end. And that is my introduction to genetic engineering.